In this first profile on this channel, we are immensely happy to talk about our first modern explorer, Professor Jane Clark. Professor Jane Clark is a professor of molecular biophysics, born in 1950. Her pioneering work helped us understanding protein folding and misfolding. She is also the president of Wolfson College, Cambridge. 20 years of her research work to understand the protein structure explained us how proteins form folded structure and what is the significance of it. It further made a way forward for a new level of research with the help of new powerful microscopes. To understand the importance of her work, let us understand what proteins are and what role do they play in human bodies. Proteins are a class of nitrogenous organic compounds which have large molecules composed of one or more long chain of amino acids and are essential part of living organisms, especially as structural components, body tissues such as muscles, hair, etc., and as enzymes and antibodies. Every cell in human body contains protein. The basic structure of protein is amino acids. These proteins provide many essential functions in our body, such as digestive system, then muscles, contraction and movement to support the body, antibody support immune function, and support the regulation of and expression of DNAs and RNAs. So how protein works? Proteins work as shown in the slide. DNA's transcription plays into RNAs. RNAs translate the instructions into proteins, and proteins actually give rise to a phenotype, that is a particular object's defined body structure in that body. The basic structure of protein is a chain of amino acids. Amino acids are building blocks of life. Chain of amino acids form primary structures, then connects to form secondary and tertiary structures. These forms then create quaternary structures, which are the functional proteins in our body. Now, protein folding is the physical process by which protein chain is translated to its native 3D structure, typically folded conformation by which the protein becomes biologically functional. Professor Jane Clark's work area for 2 decades is predominantly in understanding this protein folding structure, asking question why, how. Amino acids interact with each other to produce well-defined 3D structures. Failure to fold properly produces inactive or toxic proteins that malfunction and cause a number of diseases. Several neurodegenerative and other diseases are believed to result from accumulation of amyloid fibrils formed by misfolded proteins. Many allergies are caused by incorrect folding of some proteins, because the immune system does not produce antibodies for certain protein structures. Jane started her career as a science teacher, working in multiple secondary schools, mainly in Northumberland Park Community School as a head of science department. She got continuous encouragement and guidance from her mother. Opportunity knocked on her doors when she got a chance to study in America while going along with her working husband in America in Georgia Tech. She took the opportunity to study the proteins. She started her scientist career later in Cambridge for another 20 years. Her fundamental studies revealed the presence of both parallel pathways and frustration in the energy landscape of apparently simple proteins. Most significantly, she has made very important advances in the study of multi-domain systems. From her discovery that aggregation and misfolding are determined by sequence similarity, through to her seminal studies combining force spectroscopy and protein engineering to elucidate mechanical unfolding energy landscapes. She has transformed our understanding of the evolution, folding, and energetics of multi-domain proteins. This work helps human understanding of genetics of certain diseases and disorders. She pioneered the application of protein engineering techniques together with single molecule force spectroscopy and simulations to investigate the effect of force on proteins. She has authored more than 100 research papers. She was awarded the Biophysical Society of US Genomics Award in 2010. She was also elected for as a Fellow of the Royal Society in 2015. Her qualities like adaptability, continuous readiness to learn, positive approach to convert a turn in the path of life to a positive change, and perseverance has immensely helped her achieve well-deserved praise. She continues her research work to understand disorder in protein structures. She is indeed a modern explorer for the humanity.